Okay, welcome everybody of you for our today introduction to molecular structural biology. I know that this is a bit unusual setup that we have this time because of the corona crisis, but I hope that you get some insight. When you want to make a question, yes, after some time, it would be wise. Valerie has a microphone on or can switch it on. When Valerie is making a look onto the public chat, yes, and making maybe some mark if somebody wrote some question, in the public chat so that I can answer it. Is it okay with you, Valerie?
Yeah, it's okay.
Okay. So, okay, molecular structural biology. I guess that most of you, or all of you, are chemists who have had some education in basic chemistry, who also have mostly some general background in biochemistry, molecular life sciences. However, I do know that, not everybody of you has been attuned to the same kind of level. So what do I expect in this lecture from you? So from you, I expect that you know at least the basics of biochemistry. So when you don't know what are the basics of biochemistry, okay, when you have listened to the BC1 lecture, that is the basics, maybe that's already, that's really enough, yes. When you have heard BC1, it's fine. When you know basics about translation, transcription, and DNA replication, and what an enzyme is, and what amino acids are, I think also that's fine with me. If you have no know-how at all, yes, of biochemistry or anything related to that, this lecture may be a bit hard for you because it's called molecular biology. You should have a general understanding, And in case you are not certain, I recommend the Stryer Biochemistry textbook, which is an excellent textbook, which is available in most languages nowadays, and really a good resource. Or the Food and Food, as we have used it also. In this lecture, we will mostly refer to the textbook. Let me just make here my laser pointer on. I want to have a laser pointer. Where's my laser pointer? Okay. So that we can use this textbook here, textbook of structural biology, which is really quite useful. And I think I put an electronic copy also on our Ilias resource, So it's not the book I would like personally as a textbook, but it's at least the closest one we do have, yes, in terms of this kind of lecture. There are also some other textbooks, many other textbooks. For example, I have here shown here Tamara Schlick's book about molecular modeling and simulation. It's pretty interesting for people who are more interested in the computational aspects, yes, of modeling of biostructures. Or, there are also some people who would like to know a little bit more about protein crystallography, how structures are made,
Excuse me, Professor.
Yeah. So today I will give a very general intro, but two fancy examples. Then second, I will show you, or we come to the general point for today. Conformation is determining everything, in terms of biomolecular structures. Third, I will come about the notion of the domain. And finally, what is our current status about the knowledge of biological structures, and some personal comments. And questions, as I have indicated, please ask them. We have Valerie as let's say, as a question co-moderator, and also as a colleague who is looking, that we are not getting completely out of control. Okay, so let's look to point number one. So all of you know that actually all our life is based on nucleic acids, and genome information. Genome information means that we have gene sequences, coding gene sequences, which are under control of regulatory regions, which code in most cases for proteins. These proteins are defined, of course, by the collinear protein sequence. Now the main point is here. Protein sequence determines structure and it also determines biological function. So a biologist would be very interested, or in most cases very interested, what is the function, for example, of my protein X, what is it doing? Is it doing some signaling? Is it having an enzymatic function? Is it in some metabolism present or not, that's a major point, when you ask about function. For what is it good for? Yes, what is it doing in the organism or in the context. However, on the other side, as a chemist or biochemist or a structural biologist, many of us are interested, how is the function actually achieved? How is it achieved? What is a mechanism? And I don't have to repeat how important mechanisms are for chemistry. It's our basic part, to understand the reactivity of compounds. And it's the same with biomacromolecular structures, and with biomacromolecules, From structures, we can mostly learn quite a lot about the potential mechanisms. However, in some cases, we also learn, just from structures for what they are good for, what is their real function. For example, you have a gene sequence that is not homologous to anything that is known so far. You can translate it to the protein sequence, but it doesn't tell you more. You make a structure and you don't have a function. You make a structure and then you see, it is related to for example, to a domain that is having some enzymatic function. It has active site residues, but the evolutionary distance to the before known enzyme is so far that I couldn't see it on the level of the gene, so that I can predict it's an enzyme, and then I can ask a specific question, what is it good for? So what you have learned is there's a relationship between all three parts here, between sequence, function, and structure. So what do we want to achieve in this model, in this BC5 model? Very general. We want to learn in general how we can approach these complex biostructures. You see them, they are complex, and when you look on them roughly, you do not get a lot of know-how. It's much simpler when you have benzene or benzoic acid or orthohydroxybenzoic acid, yes, and then okay, that's a salicylic acid, it's a simple compound, but that's not the case with biomacromolecular structures. Then, second point is, to do the first one, it's very useful to recognize the most common structural motives. We will define what a motive is and what a structural motive is in terms of biomacromolecular structures. Then, how actually are these structures made? We will not go into depth for this. There's a module available, in the winter semester, where you can learn protein crystallography from the basics. That is offered every winter semester. But we will more or less only go on very general aspects, of how these different methods for structure solution work, what their advantages and their limitations actually are. And then fourth, also a very interesting aspect, what is how can we add up understanding to our protein structure, some biophysical understanding, for example, for protein-ligand interaction or protein-protein interaction. To do this, we will have some practical things, here, as you see here, we have some practical things, and for this we use the computer, yes, For example, we will be able, after this module, to predict protein structures in many cases. We will be also able to predict, we will do some docking experiments, to predict some protein-ligand complexes. We will be able to correlate the sequence room to structures to get more out of this information with the complementation of many sequences which are available. And finally, and last but not least, and I think that's the most obvious thing, also how to visualize them, for example, to make some, to really get a good view how they look like, yes, and maybe even make some movie. So hands-on experience is important here, yes, and I hope this is also the thing that for most of you is also of the largest benefit of this lecture. So why do we make protein structures, or why do we make any kind of biological structures? Clearly you can say, naja, because I want to become famous, I want to get a Nobel Prize. Yeah. When you look into the history of the last 25 years, the Nobel Prizes for chemistry, I have not the current number in mind, but I think it was 8 or 9 Nobel Prizes, already only for work, on biological structures. May it be either, for example, the Nobel Prize to McKinnon for the work on ion channels, may it be to Kornberg, the Nobel Prize, for his work on RNA polymerase, to Hartmut Michel for on membrane proteins, and so on and so on. Or I think 2 or 3 years ago, to Richard Henderson from Cambridge for making cryo-EM a method with which we can come now really to molecular structures. So that would be something very personal, yes, to become famous, yes. But I think there are some scientific issues, yes, which are at least as important or more important, why we are interested in making structures. So when you look here on a structure, clearly, you can do something to recognize a fold. As in the example before, we had it, correlating sequence with structure and maybe get some function out of this. This can be done by using evolutionary relationships, yes, and to study them in detail. Most of us are mostly interested in more or less like ligands, protein ligand complexes, yes. For example, we have now a huge interest in compounds that bind to some viral proteases, for example, or spike proteins or whatsoever. Yes, you know what I'm talking about. These ligands may be potential drugs with which one can fight them, or these ligands can be from some enzyme, for some enzymes, some substrates or products to study the mechanism. Very interesting. It can be also, that we would like to understand more or less very complex multimeric biological complexes. Some proteins are not existing, or also RNA, ribonucleic acids, are not existing on themselves. They form complexes, and very large complexes. We can now study them, especially with cryo-EM, and to see their relationship with each other. We can study mutants. Mutants are very important. Think about, for example, a disease-causing genetic defect by a single site mutation. A very good question is, why is it then the protein product out of order? What's the reason for this? We cannot see this usually from the sequence. Also some other issues here, like more biophysics, something about shape, electrostatic properties. Very interesting when you want to predict, for example, binding sites, or want to do in silico screening, and also here mechanisms, catalytic clusters, and so on and so on. So there are many, many reasons. But before talking all the time by myself, coming from this point, maybe just the last point before I make the talk over, just to remind you about length scales in chemistry and biology, and you should know them. You are chemists, yes, and you should also know them, what these length scales mean for the biology, yes. So when you are in the angstrom region here, the atoms, 10 angstroms, drug molecules, yes, from, let's say, 30 angstrom to a few hundred angstrom, polypeptides, ribonucleic acids, yes, 100 nanometer, the 1000 angstrom about this range, yes, or a few hundred angstrom. Then we come in the micrometer range, bacteria, and then we come in the range a few micrometers to several tens of micrometer cells, and this is a kind of scale, from the initial compounds. But before I'm talking now, I hope that you will get also the sound. No, it's still not. Yes, I should make a look, on the left screen sometimes, yes, because it's telling me. Maybe also some about the difference, yes, This is not a biochemistry lecture, yes, In a classical biochemistry lecture, you have heard them, yes, biochemistry 1 or 2, where we are talking about, about, for example, a metabolic pathway. Or when you are going, for example, to the biologist and, for example, to molecular biology lecture, yes, where you learn something about signal transduction, you also get this kind of things. These are all paths, yes, these paths, moment, no, right. These paths, hopefully it goes on, otherwise, these paths, give you something about the relationship between the compounds and the enzymes, what is converting into what, and also this kind of signal transduction paths. But this is not the kind of structure, well, what's interesting for structural biology. We are interested how this rough is interacting with this MEC in the phosphorylated state, yes, or how actually we have to understand that it can do so when it's activated by a RAS protein that is carrying a GTP. What is the difference between the RAS with GTP bound and with GDP bound? These are things, we want to understand. The relationship that these interactions, are something that is being from the molecular biology. And the same is also with the chemistry and the enzymatic mechanism. So classical biochemistry and molecular biology is mostly defined by relations which are given by chemical conversions, interactions, and turnovers. But now to the little interruption, to the break, and I hope that we get the sound here, because I do not know it's running. Oh.
The World Health Organization was informed of an outbreak of pneumonia of unknown cause detected in Wuhan, China, on the 31st of December 2019. Today we know that the infectious agent was a virus of the coronavirus family, which was named SARS-CoV-2, that causes a respiratory disease termed COVID-19. This virus has not only received widespread media coverage, but also caught the attention of scientists who have been busy analyzing the virus, the disease it causes, and how it spreads. There is already an enormous amount of information available about the virus, and that is why we thought to summarize some of the information in this video.
So, I interrupt just at this point. You can look to the video on YouTube by yourself later to get the summary about the coronavirus, which I think is pretty interesting. It's a very nice demonstration, about, how we approach nowadays this kind of phenomena. We have seen structural biology plays an important role at the moment, yes, because there's a worldwide run, for getting compounds against this virus, also to get some of these viral components, structurally defined, and also not only structurally defined, to make predictions what may be the proper targets, So as it was just a reminder, what is actually the broader context in which we are working. But, I want to talk myself and not give you the YouTube video all the time, just 2 minutes. Look by yourself a little bit later. I want to go on, about our understanding of life and what it has to do with the molecular structures which are underlying it. So here you see Escherichia coli, a bacterium from our gut. About a 5,000-fold magnification. Here you see the flagella, with which it can move through the medium. You see here the the 2 membranes, around it. The pili, is a structure model. This is not an experimental structure of a colobacterium. It's made by assembling all the available structure information into this single picture. Actually done by David Goodsell in this fancy book, Wie Zellen funktionieren. You can find it in the literature folder of our Ilias resource. And make a look in it. It's really well written and well illustrated. And then you see here in the medium in yellow, these are the nucleic acids, and in bluish, these are the proteins around it. So when you look on it, it looks very complex, but there are some general structures to be present. And one can go a bit closer to it now, to understand to see what these components actually, what their context actually is. The first point is, everything that has been shown before on the slide with arrows interconnected with each other, yes, converting this to that, yes, and then followed by a subsequent turnover to this, is the kind of way how we do the abstraction, of all these kind of interactions that are happening at the same time. There are important interactions which are really existing, and there are non-interactions. Non-interactions are interesting because most proteins have actually evolved not to interact with anything else, just with the partners they really like to interact with. That is something, that is negative design. So here you see many components, and I will give you some brief overview about these ones. Maybe the first point is, when you look here, this is really, really crowded. It's absolutely crowded. That means the concentration only for the proteins inside cells is more than 100 milligram per milliliter, a tenth of a gram per milliliter. And proteins are a bit fluffy. So you have to imagine that, yes, when all of these proteins would go out of solution, you would have a cloudy, whitish mass with a lot of water attracted to it around, So this is actually how it looks like. It's densely crowded. These macromolecules occupy up to a third of the whole volume. So they're not free floating, they tumble around, they cannot diffuse rapidly. Some of them are bound maybe to larger structures here like the DNA or membrane bound. The degree of motion and of interaction is time limited. So when you look here, this is, I think, a 5 million fold magnification about, yes, from the model. It's not an experimental structure. It's everything what Goodsell was modeling according to structure information available. You see here the typical environment, here this is probably some enzyme, from the glycolysis. I don't know whether it's GAPDH or some other glycolysis enzyme. Here, we see some nucleic acids probably here around, curling around. Here, these dots, these small ones in whitish are water molecules, which are filling up everything. Some ions here in red and in green, cations and chloride ions, and some phosphates, and some compounds, some metabolites in between. So that's about how that is to be expected, how it looks inside cells. So it's a crowded environment. And the funny thing is, when looking on something like this, and this is really something for which Goodsell became famous for, yes, for making this kind of fantastic illustrations from macromolecular structures, gives you some impression how integrated these whole systems are. This is a simple bacterium, nothing unusual, but you see, how many different components are inside the membrane. For example, when we go here, F is, I think, some pylene structure. Yes, P is a transporter that is transporting out compounds which the cell does not like. Yes, it's unspecifically transporting them out. It's ACRB. Then we have here, the peptidoglycan layer here shown as H that is going in between, and some enzymes associated with it. For example, like beta-lactamase, the target of our antibiotics. Then we have here some enzymes. We see some more enzymes. Some more enzymes are in here. Here's some receptor, I guess. Actually, I have to look into this kind of description. Yes, some enzymes. Magnesium transporter. Yes, all the kind of things. And here, here shown in bluish. This is MIEB, that is an actin-like filamentous protein that is giving the bacteria their shape. And it's also of interest, for example, for groups like Peter Graumann's group, who are working on bacterial shape and cytoskeletal components, which are involved in giving these cells structure. Here in this kind of case, in this kind of part of the figure, we see all kind of proteins which have something to do with DNA, with transcription and with translation. Here, for example, here in yellow, as you see here, there should be something like DNA, double-stranded DNA from our genome. Here's an RNA polymerase. This one here projecting is the RNA. Here's the ribosome that is translating it to a new polypeptide. Here's some tRNA approaching this ribosome. There are also some other components here. For example, that looks like something that is sucking this up. It's a proteasome-like protein called ClpP that is degrading proteins in the cytosol. We have some chaperones there. What is the number of the chaperones? That is K. Where with K? No, K is ClpP. Oh, maybe something going around here. Or it's GROY A. It could be GROY A. No, this is a proteasome. Yes, the ClpP. It also looks similar to K. Yes, this one is degrading, and this one is helping that the proteins are folding. Yes, so you see everything at the same time present. So here, for example, the machinery, yes, that has something to do with DNA replication and DNA maintenance. Here you see many components. For example, the DNA polymerase, here's a topoisomerase, that one we had before. That looks pretty conspicuous to be that is MUT-M, this is a DNA repair system. This is a replicase here, B, yes. B is also polyisomerase. Where is A? No, where is A here? A, I don't see A. Now you see I'm now confused by these many letters and these many components. And you hopefully are confused as well. And here's something, but I hope it's quite, no, we are not over here. These are enzymes from just A is everything enzymes from glycolysis, and B from the citric acid cycle. Remember, it's a bacterium, yes, This is not compartmentalized like eukaryotic cells. The enzymes or the glycolysis are in the same area as the enzymes for the citric acid cycle, so they are present at the same time, yes, and just being regulated, of course, by post-translational modification, which is important because they are just neighbors, yes. We have to regulate them, that they are not making some futile cycles. And here at the end now, I think that's the last slide from all this multicolored, super fancy stuff, Here's the flagellar motor. A is flagellin, yes. B are the MOT proteins, which are embedded in the inner membrane. C are the protein components which make the rotor. For example, Gerd Bang is very interested in the flagellar motor. This motor is regulated by key Y. This is here, this small little protein is either phosphorylated and goes into the MOT component, or not phosphorylated and doesn't go there. And its phosphorylation is controlled by this chemotaxis receptor. This is a receptor that is feeding aspartate here at its binding domains and transmitting this to a signal. Here we are an adapter protein and a protein kinase to finally key Y. So this is the kind of components for from chemotaxis in bacteria. Pretty complicated. For example, Viktor Surgic at the Max Planck Institute is interested in chemoreceptors and the kind of integration and kinetics, and also the possibility to construct novel kind of chemotaxis receptors by making hybrids or predicting novel affinities or specificities for them. So this is the overview I wanted to give about Escherichia coli. Now let's go to the very, very, very basic things back. So we fall now down from the heights to the depth. So all proteins, as you know, consist of amino acids, and our 20 proteinogenic amino acids are all L amino acids. You know the kind of different representations, your Fisher projection, chemists like this, This kind is a ball and stick representation. You can use, it's useful, for example, for our molecular viewer programs, or this is a space filling representation that gives you some idea how the space is filled out, but you do not understand the structure and the chemistry behind this. So this is more useful. This is for the chemistry, the most useful one usually. But this can be more useful, this representation when you look on the structures. So when we consider 20 different amino acids, one of the first questions is not knowing the single letter code, as you should know it, yes, learn the single letter code when you have forgotten it, yes. L like leucine, A like alanine, G like glycine, valine, isoleucine, proline, F is phenylalanine already, yeah. Okay, so when you look on these 20 amino acids, they are not evenly distributed in the proteins. So you see there are amino acids which are very, very common, like the leucine or the serine or the glutamate, here between non-polar uncharged and charged polar, yeah, and amino acids which are very rare, like tryptophan, very complex amino acids with the indole side chain, or the one with the cysteine, very important amino acids for many enzymes, also for disulfide bridges and so on, but it's very rare, yes, because it's also metabolically very expensive amino acid, in contrast to something like alanine, which is very simple to synthesize, just from glycolysis in 2 steps, or glycine also in 2 steps from a glycolytic intermediate. Very simple, but we have also rare amino acids. And one of the points is, what are also the rare amino acids good for? And we can usually, or histidine is also a rare amino acid, and we can explain them, in the structural context. Mostly we find that many of these rare amino acids are highly conserved in structures because they have to fulfill either certain structural functions or mechanistic functions. You can imagine cysteine and histidine are prime candidates for residues in an active site, for example, because of the catalytic potential. So there is some correlation with degeneracy of genetic code and the length of the biosynthesis pathway. Clearly, as rare as longer it is. Now, these proteins, these amino acids, when we go, and even the simplest amino acids, when the structures are different, the configurations are different for the 20 amino acids, but even a given amino acid, like glutamate, can exist in many different kind of states, which are defined by the different conformations. The conformations are defined by the torsions. You are chemists. I hope I don't have to explain to you these kind of basics, like gauge minus, gauge plus, trans, Newman projection and these kind of things, yes, and remembering, trans, gauge minus, gauge plus are the stable conformers here, for example, for butane, but staggered ones, here like 0, 120 and minus 120 degree here, for example, this model compound are the energetically disfavored conformations. So biological structures are mostly determined by the conformational isomerism of their constituents, of their amino acids. Let's go step by step when making the point strong. Let's go here, torsions of the peptide main chain. Yeah, you remember the peptide main chain. We have is the alpha atom from amino acid 1, now from amino acid 2, sorry. Here is the amino group, and that is making a peptide bond with the preceding amino acid number 1, here in this case. So there are 3 different angles that have to be considered. One is the torsion angle phi, around which we can rotate here. This is corresponding to phi, by which we can rotate, almost freely rotate. We have the torsion angle psi. These are the Greek letters for this, around which we can also freely rotate. So there's also as you see here in this kind of drawing, also shown as a single bond, the peptide bond itself with the angle omega. Now, omega is special. Omega cannot adopt all kind of values, It cannot adopt, for example, usually it cannot adopt. There are always exceptions, but it can usually not adopt, for example, 90 or minus 90 degrees, yes. And the reason for that is given, as you hopefully remember, by the peculiar character of amide bonds, Amide bonds, can be described, by several mesomeric forms, this one or this one, and this is somehow in between, indicating that you have a partial double bond character, for this kind of peptide bond. So a partial double bond character as also reflected by bond lengths, yes, and here recognized by Linus Pauling already in the 40s. And here, this book I had found when I was a boy in Deutsch, I think, yes, even in our library. It was a very impressive book, really. Very interesting how he wrote it. Much better than nowadays books are written, yes, in terms of the language and the kind of insight that we have here, partial double bond character. So that means, between all the other kind of things, with dipoles and so on, that actually this bond can exist when you remember like 1,2-dichloroethane, yes, either in a trans or in a cis configuration. So when you have a trans peptide bond, yes, like this one here, the C alpha atoms are opposite of each other of the peptide bond, and they are here in one single plane. This atom, this atom, this atom, all atoms are in one single plane. This is a transpeptide bond. 99.95% of all peptide bonds are like this. One can also remember, quote now the number here, 3.85 angstrom. That is the distance between this C alpha and that C alpha, and to the next C alpha, and to the next C alpha, and to the next C alpha. Yes, with all transpeptide bonds. It's always the same distance. The cis peptide bond is also existing. Here omega is in 0. Here the distance is about 3.1 angstrom, much closer. It's rare because usually these kind of substituents can collide here, so it's energetically for most amino acids highly unfavorable, except proline, and you see all the other 6 are in one single bond. Okay, so that's about, omega. 2.9, sorry. Yes, okay, and here's something about except proline. Proline is special, as you remember. So we have along the polypeptide chain only a limited conformational flexibility that is defining our macromolecular structures. Remember, that is different, when you go to polymer chemistry and you deal with many kind of polymeric compounds which are also of technical use. Many of them have more degrees of freedom, yes, and form very stable structures, mostly because they can form crystalline like local domains. Here in this case, we have already this almost invariant omega bond, yes, omega angle here, and we have these two angles phi and c, and as you can imagine, they can also not adopt any kind of value. They are also limited because otherwise everything would start to collide here in the structure. So when you plot phi versus psi, remember, this is biochemistry 101, you get this Ramachandran plot, and you find here a large region here for parallel or anti-parallel beta sheet-like structures. Yes, it's here. Or this is a region which is allowed where you find alpha helical structures. Yes, it's defined there. And in this region, you find there some kind of turns, yes, which have some alpha-like left-handed characters, but which are very short, But everything plays in these 3 regions here, for L amino acids and typical proteins. Nevertheless, what we have seen before was the average of all amino acids. When you go for an individual amino acid, they are highly different. What kind of oligomer or what kind of combination of phi and psi they can actually take. For example, glycine, yes, the only non-chiral amino acid, you see it's symmetric. That forms also a large area here and there, yes, but it can also be here somewhere, yes, So you can predict that would form perfectly this kind of loops stuff, yes, when you have left-handed helical requirements, maybe. It would fit perfectly there. but not proline. Proline is a ring structure, yes, it cannot change its phi value. That is almost fixed here, yes. That has to be just in this kind of small region. And therein, it's like the 18 other amino acids, the typical one, with beta, alpha, and the other area there around. So that's what we can know from our kind of main chain structures and the kind of conformers that can be adopted along phi and psi, giving rise to this kind of secondary structures. So that's fine. Is there more to be learned about torsions? Yes. We have side chains. Side chains are sticking at every position apart from glycine, or let's say maybe alanine, yes, because they are so small. But all the other 18 amino acids, they have some side chains, yes, And these side chains, yes, also take up certain kind of conformations. They can be also rotated around the single bonds, like here, here, and there. This is the amino acid glutamate, glutamic acid. It has 3 rotations around which one can rotate Chi1, Chi1, Chi2, and Chi3. Sorry again for these Greek letters, yes. People like Greek letters very much, Chi in this case, yes. Now you can make a prediction yourself. Can there be any kind of combination of all these kind of values, or has there to be some restrictions? Of course, there have to be some restrictions because, as you can imagine, otherwise you would bump in here, for example, the side chain maybe to the main chain. There have to be some restrictions. And these restrictions can be defined as so-called rotamers. So you do find for all these amino acids, for at least these 18 amino acids or 17, when you also subtract proline, yes, that they are having not arbitrary combinations of these chi angle values for the side chains, but they have preferential combinations. You see this, for example, here for the amino acid tyrosine, yes, the aromatic amino acid with this kind of hydroxy group, this phenolic group here, yes, You see, there are mainly 2 kind of combinations, for chi1. The one shows in this direction, the other one shows in that direction, yes, for chi1. And for Chi2, there's also a certain kind of flexibility, but also not so much, because otherwise it would collide again here with the main chain. And that's also true for this kind of conformer. So what one can predict is, and I think that's also found by structural biologists, when you solve protein structures and you make them at higher and higher resolution, you do usually observe that the side chains fit even better to the theoretical rotamers you got from database analysis, than when you were starting with low resolution structures where, let's say, experimental error is still a problem, to define the precise conformation of a side chain. So rotamers are element of a set of conformers that arise from restricted rotation around a single bond. Okay, there's even some definition for this. So what does it mean? Here, for example, all this kind of different amino acids, arginine, asparagine, and here this kind of combinations, Chi1, Chi2, Chi3, Chi4, and you see, there are just a few for many of them. So a handful of conformations. So that tells us that even when you consider I have 20 different amino acids, and I have even some variability around the main chain, it's not an infinite variability I do have with these rather bulky monomers with which I assemble my proteins. So let's also give some words on secondary structures. Yes, I think I have to do this again. Yes, secondary structures are repetitive local structures which have the characteristic that they have the same phi and psi bond torsions. Yes, the same phi and psi torsion around the main chain. It started, with this kind of X-ray diffraction images from Asbury and colleagues, in the 30s, which were using Kerr or Wool and could observe by X-ray diffraction that there are defined spots indicating that there's a superstructure inside here, and that has been later solved how this looks like, the hair and many other fibrous proteins have consist solely of alpha helical structures, here predicted by Pauling and Corey, alpha helical structures where every peptide bond is making a hydrogen bond with a different peptide bond here. Every peptide bond from amino acid I, for example 4, is making a hydrogen bond with I plus 4, in this case 8, yes, and that it's done for all of these main chain atoms. So that is a regular moment, for alpha helical structures. And I think, it's a bit repetitive here now, but I just would like to highlight it because it was taught in Biochemistry 1, but not everybody heard to Biochemistry 1. We know the height about it. 5.4 angstrom we require to go over one helical turn. Yes, we need 3.6 amino acids. That was a trick, how they solved it and not others before, by making this model. There's not a 3 or 4 residues required, just 3.6, and it can go on. There are some conclusions out of this. Yes, for example, you have a large dipole moment. Yes, along an alpha helix. At least when it's an apolar medium, then you have a very large dipole moment. Here, for example, in this cation channel KCSA, yes, these kind of helices are directed to the central potassium ion to stabilize it energetically so that it does not feel any kind of energetic difference from being here or outside the membrane. Here it's inside the ion channel protein. So this is a motive that is making it almost frictionless transport possible around ion channels. That's interesting. It's also interesting that when we consider our 20 different amino acids, the propensity, that means the likelihood of being found in alpha helix is different. For example, a proline is not an amino acid that you would like to have in the alpha helix. Remember, it has a cyclic side chain. Yes, it's making steric interference with the neighboring residues. It does not fit in. It fits only, at the beginning or at the end of an alpha helix, but not in the midst of it. And the same is true for glycine, just for the opposite reasons. It has no side chain at all. It's pretty flexible, and in this way is not stabilizing any kind of secondary structure on itself. However, there are some amino acids like alanine, which really like to be in alpha helix. Also arginine, some of these larger, bulkier amino acids like to be in alpha helix. There are also some other ones which are similarly bulky, but they don't like to be there. These are all amino acids which are branched, near the C beta atom. That means in the side chain, and the branching causes also interference with the main chain. So those ones also don't like to be so much in an alpha helix. So there's a different propensity. Here, helix breakers, proline over glycine, histidine, asparagine, tryptophan, valine, isoleucine. These ones are all branched at the C beta atom, and these ones have some functional side chain at the C gamma atom. When we look perpendicularly on an alpha helix, and now maybe I switch now, sorry, to explain it on the keyboard. Sorry, I have to switch it now. I hope you are still there. Yes, I see now you that you're still there. quite good. Let me just try whether I can get it. So when this is an alpha helix, yes, I'm not the best writer. Yes, this is an alpha helix. Alpha helix, yeah. Here's the N-terminal end, here's the C-terminal end. The side chains project, like from a Christmas tree. We can also make a view, perpendicular to it, yes, and just look on the helical axis, yes, and ask ourselves, how is the distribution of the amino acids when we look to this helical axis? Now let's go back to our PowerPoint. So this helical wheel presentation. So here you see this example of a helical representation. Here amino acid 1, and then we come to amino acid number 2. Upsala, I have to switch on the laser pointer. Laser pointer went off. to amino acid number 2, 3, 4, and so on, you know. And the helix is going into your direction, yes, you know, when you're now looking on it, you know. So you see here, for example, that around here you have all kind of hydrophobic amino acids, leucine, 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 leucine. Here maybe some tryptophan, no leucine. Here you have the hydrophilic amino acids. So this is a so-called amphipathic helix. Amphipathic, how is it written? Yes, you know. Amphipathic. Let me just get back to the drawing board. Hopefully it writes. Amphipathic. So that means from the one side it's hydrophobic, and from the other side it's hydrophilic. There could be, for example, a helix that is, for example, laying on top of a membrane. When you have a lipid membrane, you know, and that would be then a helix that would lay on top of it. Yes, you know, here it would be hydrophobic. Yes, here's the membrane. And there it would be hydrophilic, yes, you know, or the other way around, yes, you know. Here it would be hydrophilic, yes, you know, because here would be all water in like what's around. But in this case, I have just shown to you now, it is probably the helix of a leucine zipper-like protein, or of the keratin itself from the hair, because it forms some called coil-like structures by which the kind of hydrophobic regions are actually making the common interface, and these kind of amino acids then intertwine with each other to make a coiled coil. So, are these the only helices existing in nature or in biomolecular structures? No. Yes, you know, it's the most common, the alpha helix that we have talked about. You know, here in this nomenclature is exactly defined how the structure is actually generated. You require 3.6 amino acids for one turn, and you have to go over 13 bonds. One of them is a hydrogen bond, you know, to close the cycle. So when starting here from the lower part, I have to go over 12 bonds, and then finally I have to go over the hydrogen bond to close the cycle. So this is the right-handed alpha helix. There are also other helices existing in nature, in macromolecular structures, however, only to a small percentage. One of them are these 310 helices. You require just 3 residues. You just require 10 bonds to close the cycle, and you see that it is slimmer, yes, this helix, you know. It is not so broady as alpha helix. And there are also some so-called P helices existing, you know, 4.4 amino acids, 16 bonds, you know, that is taller. Mostly only very short regions are found in structures. These ones, you know, the 310 helices sometimes can be found to some longer term, to some longer extent. There also exists, of course, the way, you know, of defining even the shorter one, the 2.27 ribbon. Yes, you know, this is, however, actually a structure element that is actually getting us, you know, to a different kind of secondary structure, because this is usually found in beta-like structures, in beta ribbons. So beta sheets, you remember biochemistry 101, anti-parallel, parallel, very similar, side chains top down, or here top down, and always alternating, you know, and they have an intrinsic twist, you know, and form then these twisted beta sheets, each beta sheet consisting of several beta strands. This is a porine, it's like a barrel. This here is from some bacteriophage, some viral-like entity, you know, that is infects bacteria, you know. It's a right-handed twist here with a single alpha helix. It's a bit more complicated structure, yes, you know. Interestingly, getting back to our issue, we have amino acids, we have side chain rotamers. What about those ones? Is there any kind of, let's say, interaction or let's say correlation between this secondary structure that we do observe, alpha helical, beta, and the kind of rotamers that can be adopted? And this is indeed the case, yes. So secondary structures, have some influence on the kind of preferred side chain conformation on their rotamers. Yes, they like to adopt. For example, you see here in green, yes, this alpha helical stuff. Yes, that is here the continuous line for, let's say, glutamine. Yes, tuck, 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 60 degrees, 180, 300 degrees, Gorge minus, trans Gorge plus. But you see that there's already some effect when you go here to the red one, to the beta sheet structure. Yes, the Gorge minus is preferred over the other ones. And some others show even stronger effects, yes, on this kind of rotamer distributions. And that is interesting because it tells you also one of the problems, that we do have. There's a context-dependent preference for local conformations of side chains. Many of you know that we nowadays like to use high throughput computing in order to simulate complex macromolecules, for example, in molecular dynamics. So in there, we have to describe everything by force fields, but these force fields are usually predicting the same kind of energetics for every glutamine, yes, So not taking into account, that actually that may differ locally depending on the kind of secondary structure they are being part of it. And that is causing that some of these force fields, are only pretty pissy, yes, of reproducing this kind of rotamer distributions. This is an ongoing research project by these groups who developed these methods. I think the KN1 force field 19 from ENBA from 2019 has now considerably improved about this. Force field 14 was quite good, So that's something, where actually molecular modelers are still making some progress to getting better predictive tools. But this is a general problem. Local effects have some influence on the energetics. Also, when you consider biochemistry 101, alpha, beta, maybe some terms, everything looks so perfect, but nothing is perfect in reality. For example, when you deal with beta-like structures, you see here the common deviations, yes, from this very regular patterning field. Here you see the so-called beta bulges, This is a classical anti-parallel stuff here, but you see there's something inserted, something distorted here, yes, Here a bulge, there a bulge, here goes something in a parallel beta sheet around, This here also some other distortion, Usually you find at this kind of positions highly conserved amino acids, because for example here, a glycine could be the case, that here you have this kind of deviation in, When you analyze, for example, sequence variation and wonder why is this amino acid glycine always conserved, I guess you would be well done when you have a structure available that you can make a structural prediction why this amino acid has this high degree of conservation at this particular position because of this kind of irregularities which are built in to form the different kind of structures. Yeah, and then there are other kind of structures existing, but I flipped this over, so proline stuff, and collagen triple helices, yes, Yeah. At the end, they are dictated, of course, by the size of the glycine and that every third amino acid has a repetitive motif. They're also part, these collagen triple helices of many viral stocks, not now of corona, yes, before you get it wrong, yes, but of other viral stocks, because they are really pretty stable, the structure, and at the end they have then maybe the receptor domain of the virus sitting or the enzymatic domain sitting to which with which they can make some interaction, but not only viruses. So we come close to the end now about the secondary structure stuff. Just to remind you, all the secondary structures are connected, by turns. These turns are highly regular. Here you see a turn 1 prime. Here you see a turn 2. There's of course also turn 1 existing. They just differ, by the orientation of this peptidic bond. This one looks to us in turn 1. It looks inside here into the paper, that is determined again then by the identity of the amino acid here. Yes, glycine or not glycine, and some local interactions. Turn 2 is the same stuff here. You see also glycine at this position and a large glutamate at that position. These turns are very interesting because we have many binding proteins which use especially the loop regions or the turn regions to make very heterogeneous molecular surfaces. The molecular surface is made by a beta sheet on itself by variation of the amino acids on top of it are not so large. However, the turns, they can give up, or the loops, they can make very different conformations and present the side chains in very different ways. So many binding sites depend on the conformation of loops, Turns are the simple ones because they are regular. But when they get longer, 4, 5 more amino acids, yes, they become irregular and cannot be classified anymore because they depend on the local context. So that was about secondary structures. Maybe before I go on, would be a good idea to see whether we do have some questions to interrupt me. Yes, Public chat, there are some questions maybe. Yeah, are there some questions? Hier, somebody says, er ist kein Ton mehr. Bei mir läuft alles. Okay, jetzt hat er Tonprobleme hier. Okay, gut, ja, ich sehe schon.
Ja, bei mir läuft auch alles.
Okay, ich sehe schon, ja. Sollte man den Laserpointer irgendwo sehen? Ja, okay, gut, dass Sie darauf hingewiesen hatten. Ja, okay. Wie sieht es mit Fragen aus jetzt bis hierhin halt, ja? Wir haben noch eine Viertelstunde, dann sind wir fertig hier. Irgendwelche Fragen? Noch nichts? Keine Fragen? Oh. Sie kennen ja mein altes Live.
Ich habe noch eine Frage.
Ja.
Vielleicht eine doofe Frage zum Anfang. Und zwar haben Sie ja diese ganzen, dieses Bakterium gezeigt, wo diese ganzen Strukturen. Genau. Wie wird denn so eine hohe Ordnung aufrechterhalten? Also in der Chemie ist es ja so, dass es im Gegenteil, also gegen die Ordnung geht. Und wenn wir dieses Bild hier anschauen, dann ist das ja vollgepackt und also ich kann es mir ziemlich schwer vorstellen.
Ich gebe Ihnen, und das ist vielleicht auch, ich glaube, diese Frage ist eine hervorragende Frage, weil sie geht auch so ein bisschen an unsere gegenwärtigen Shortcomings, also unsere Probleme heran. Was verstehen wir nicht? Also die Einzelkomponenten fangen wir an zu verstehen halt, ja. Aber wie diese ganzen Komponenten da zusammen da herum wirbeln, sich da herum machen halt nicht, ob sie da lokale strukturelle Features bilden. Also das mit den, zum Beispiel die Sache, dass mit dem Nukleoid zum Beispiel hier mit der DNA, das kann man wohl noch am ehesten noch verstehen, warum sich hier der DNA-reiche Bereich sich abtrennt von dem proteinreichen Bereich. Hier ist es halt so, das ist halt ein zirkuläres Chromosom und das ist assoziiert mit jede Menge von Proteinen, die es auch kondensieren und verpacken und auch entsprechend hier so weit zusammenziehen, sodass auch wenig Platz für die anderen Komponenten dazwischen vorhanden ist. Also die Grobteilung hier, da haben wir so eine Idee, das ist so etwa ähnlich wie mit den Chromosomen auch in unseren eigenen Zellkernen halt, die auch mit entsprechenden Histonproteinen assoziieren halt und entsprechend dann kondensieren. Bloß natürlich auf der Ebene hier zum Beispiel innerhalb der Membran halt oder innerhalb des Cytosols nicht. Was zum Beispiel nicht klar ist, und mein Kollege, der Gerhard Hummer vom Max-Planck-Institut für Biophysikalische Chemie, der dortige Direktor, hatte uns auch vor ein paar Wochen erste Molekulardynamik-Simulationen vorgestellt, wo sie versuchen, in der Tat mal so Blöcke zu simulieren, wo mehrere hundert Proteine zusammen sind. Also nicht 2 oder 3 oder 4 halt, nicht, ja, wie man das vielleicht noch so machen kann halt, sondern hunderte davon, so ein Ausschnitt davon halt. Weil die Frage ist durchaus, nicht, wie kann es sein, dass eben halt es nicht falsche Interaktionen gibt? Interaktionen, die dafür sorgen, ja, dass meinetwegen hier Protein E dranklumpt an dieses Protein, ne, das waren jetzt Ribosomen gewesen, das ist ein schlechtes Beispiel hier, meinetwegen das da, das meinetwegen hier, das ist die Pyruvat-Dehydrogenase, dieser große Komplex B, ja, dass der nicht zusammenklumpt die ganze Zeit hier an A und A irgendwie daran hindert, irgendwie seine Funktion zu machen, obwohl er vielleicht nichts zu tun hat, oder hier an einem Ribosom festklumpt, der auch da irgendwie groß in seiner Funktion beeinträchtigt. Das ist nicht vollständig verstanden, weil wir können es auch nicht vollständig simulieren. Wir können es nicht vollständig nachahmen. Wir können erstmal nur Gesetzmäßigkeiten rausfinden. Ich hoffe, das reicht so als, sagen wir mal so, weiche Antwort erstmal halt, ja. Gut. Haben wir noch eine andere Frage? Das war eine schöne Frage gewesen. Ja, wenn keine weiteren Fragen, gehen wir mal in den Schluss ran jetzt über halt, ja. Nicht? Gehen wir mal in den Schluss ran über. Okay. Kommen wir vielleicht nochmal auf den Punkt halt nicht. Wir haben jetzt so Sekundärstrukturen gesehen. Wir hatten es aber am Anfang gesagt, und das wird uns beim nächsten Termin ein bisschen mehr beschäftigen, Domänen, Faltungsmotive halt, ja. Was ist eine Domäne? Muss ich hier erstmal den Laserpointer wieder anmachen. Sekunde, das dauert immer eine Sekunde, bis ich das wieder gemacht habe. Der ist leider nicht immer automatisch an. Ne, der ist immer nicht an. Sekunde. Okay, wenn wir uns jetzt hier so ein Protein anschauen halt nicht, können wir relativ leicht sehen, das hier ist eine Domäne und das ist hier eine Domäne, weil wenn ich hier das hier in zwei schneide, könnte ich mir vorstellen, das hier kann für sich selber so gefaltet bleiben und das kann hier auch für sich selber gefaltet bleiben. Also das ist schon mal eine gute Definition halt. Eine Domäne ist eine Faltungseinheit, ja, nicht? Definition A, nicht? Ist sehr brauchbar, heißt zum Beispiel für uns in der Biochemie halt, ja, wir wollen zum Beispiel irgendein Protein charakterisieren, das besteht aus 3 Domänen, aber das gesamte Protein interessiert uns gar nicht halt. Wir machen einfach ein genetisches Konstrukt, wo wir nur das hier so ausschneiden, hier, da. Meinetwegen, hier ist der C-Terminus, dann wird das entsprechende Gen entsprechend nur bis hier hingehen, einen Stoppcodon da rein halt nicht und dann exprimieren wir dann überproduzieren wir nur diesen Teil hier halt, der da ist und charakterisieren diese einzelne Domäne halt, weil sie sich von selber falten kann, nicht? Oh, sorry, I switched back to English. Sorry for this. So that would be one possibility, yeah, you know. A domain is a folding unit. One can also give more, let's say, bioinformatics related definitions. For example, like B. A domain includes all amino acids of a polypeptide chain which are nearby in the tertiary structure but not necessarily in the primary structure sequence. Give you an example, you know. Let's go to our drawing board. Yes, you know, where's a drawing board? So, drawing board, you know. So, very simple domain here, N and C terminus, yes, you know. This is really a domain, yes, you know, I think you believe me, you know. So I can say I take this amino acid here, you know, and it's in local neighborship to those ones here around. And this one here is in local neighborship to this one here. And here I also do something else. But it's not in close relationship to the residues here, yes, you know. So that it can actually be done by a computer, yes, you know, to identify what would be consistent here. That is also close. And this one is close, you know, local networks, yes, you know, but no networks here. Too far away. Distance is too far. So that would be the let's say the more, let's say, mathematical way or computational way to define domains inside a protein structure, you know, and it's actually pretty useful. So the question is, how many different protein domains, and I'm talking now only about proteins, not about RNA, yeah, about how many different protein domains really do exist. That's an interesting question, yes, I think has been of interest for many bioinformatics people, yes. First of all, we have to consider the length of a protein, yeah. The length is usually between 25 to 500. 25 is very short, yes, you know. There are some mini proteins existing, like the tryptophan cage, yes. They are as short as 25. But usually there are no domains, you know, above 250. It's only a few that may come up with 500, yes, you know. So most domains have this kind of size, 100 to 250 amino acids, maybe 80, yes, you know. So don't nail me. So these are the typical lengths for a protein domain. Second point is, how many do exist? So that is a very interesting question. That depends on the criteria you take. One of the best databases, you know, for protein domains that is partly hand curated is the SCOP database. That is in the version 203. In this database, we know about 1,200 folding motifs, 1,200 domains. 1200, which are capable now to cover about 2/3 of the genomic content. Yes, which is quite a lot. So we assume nowadays, that we already know most domains. When you look every year how many new domain structures have been solved by structural biologists, the number is strongly declining. Yes, so clearly from time to time a new domain may be found, but most of them have been already found. What do they feature? Define tertiary structure. They're compact and stable. They occur in very different proteins. That's actually very true, yes. And they're better conserved in the sequences. So you often observe, and we have experienced by ourselves, for example, with a fungal adhesion molecule, it had no sequence similarity to anything known, yes, apart from some other fungal adhesion proteins, yes, And when we solved the structure, and I was looking on it and I said, oh, that looks like a fibronectin type 3 domain, which fibronectin is a protein defined in animals, yes. It's part of our extracellular matrix, yes. So it's a very common protein fault, but one could not recognize it from the sequence. It was hidden, yes, but better conserved in sequences, And here we have folding unit structure, compact and structure and functions are evolutionarily related. That's true. So there's also another thing. These protein domains count to folded proteins. However, one should also keep in mind there are proteins which are not folded. They have that means they are not ordered at all. They are not forming compact structures, They occur maybe only in a few proteins, in a very specific proteins, yes. They are very often interaction sites of many things. These kind of disordered regions, yes, can have many functions, It's very difficult to find that out. Clearly also a case for the structural biology where it has some problems, to define, but we have no better techniques. We come in the lecture on this, And here at the end, a bon mot from François Jacob, yes. And you may also ask, are there more than, let's say, 1,600 protein domains possible? Why not? Yes, why should not be there more protein domains possible than we do find them in nature? The point is only evolution is making tinkering. It takes something that works more or less and changing it a bit, yes, But humans can do engineering, so we clearly can suppose that we should be able to engineer de novo completely new protein domains which are not found in nature. However, this process is very, very difficult to do so, because that means we are able to understand really what we are doing. And we are just starting to do that with protein modeling. Here you see some example of tertiary structures. You also get the definition. Tertiary structure corresponds to the coordinates of every atom of a single polypeptide chain. Yes, you should know the definition from the biochemistry, but here you get it again. Yes, So when you have, let's say, a file with a few thousand lines and with the residues and the atom descriptors and the X, Y, Z coordinates with this, then you have a complete structure definition of the protein just by having X, Y, Z for every atom of it. That is a tertiary structure of a polypeptide chain. That's interesting not only because of the evolutionary relationship, but because, you can come up with this kind, let's say, of diagrams, yes, as we see it on Thursday, yes, in 3D, you can simplify to these contact maps, yes. Here is a contact map shown. What is the contact map showing? Here, going from the N to the C terminus, N to the C terminus. So let's say number 1 and maybe number 100, I don't know how long this protein is. And there's everywhere a dot shown here where actually the amino acids have some contact. They are coming close together on van der Waals contact. And clearly there's a diagonal because, that's the main chain. And you even see there's some here a bit off from the main chain. Yes, here, there are some elements here. Yes, you see that here. These are the alpha helical elements here in this protein. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Because there, every amino acid I and I plus 4 are in close contact, yes, and also, of course, with I plus 3 and I plus 5 a bit, yes, So they are marked here, yes. So this is a contact map, just a two-dimensional contact map. And then you see the off-diagonal elements, for, let's say, this one is interacting with this one here, maybe with a side chain. It's maybe somewhere here in. So these contact maps usually are calculated when you have a protein structure, or you can calculate it. The interesting point about these contact maps is the contact maps also correspond to the protein structure. So in general, it's possible to transform this into that, yes, this contact plot into a structure, at least with, let's say, some predictive power. There's now since about 10 years now, a new way to come to this contact maps in the statistical analysis of sequence variation. Sequence variation. Maybe I should explain it shortly on the note here, on the drawing board. What does mean sequence variation? Let's say you have here a polypeptide. Here's, let's say, number 100. Here's number 85. Here's the many different positions. Yes, and the protein is let's say, is folded into something. Yes, maybe with a helix here around it and C-terminus. Yeah. Okay. So what does it mean? Now, yeah, just imagine. Let's take some different color. This amino acid and this amino acid here. Yes, they change. When this amino acid is changed, yes, this is amino acid I and this is amino acid J, yes, because they are close nearby and they are in contact with each other. When I change I, J has also to be changed. Just imagine, here's a tryptophan and there's an alanine. Now I make a phenylalanine at position I, which takes more space. Then J should be a smaller amino acid. Maybe it should be valine. So when I have now here, that is J and here is I, yes, I would see when I have many hundreds of sequences available, many hundreds, many hundreds, yes, maybe thousands of sequences available. And I make a statistical analysis of all these sequences and ask myself, for each position, is a covariation happening, yes? I may identify I and J that every change inducing here is correlated with changing at position J. So that is about covariation, and that is actually working now pretty nicely because it can be now used to predict protein structures from sequence space alone. Here's an example from my own hand. For example, GAL4 is a very famous gene regulator in yeasts. Very famous. Actually, one of the most common proteins have this kind of folding motifs. The structure is unknown of this protein, has not been solved yet. There are 16,000 orthologous sequences. When one is putting this into a program like Gremlin, it makes this covariation, and you get this kind of plot. Every dot here is showing here some covariation in, and it looks exactly like this protein contact maps. And the authors of this kind of program have shown that covariation corresponds closely to protein contacts, and one can actually now predict what the structure actually is here. So when one is making a prediction here, yes, one finds actually already some protein in the database, and that is shown here. This is a protein that is organizing centromeres and chromosomes. Completely different function, but it's being associated with DNA again. Yes, it's having the same fold, and these kind of transcription regulators just have taken over this fold of the centromere organizing proteins. So one can, by this kind of convolution maps, predict structure, which is really something, that is being made available by the huge number of sequences. Now at the end, the protein database, we make a look on Thursday again, but you can already make a look by yourself a little bit, I recommend this to do so. Yes, here the molecule of the month. Now I think it's corona, yes, clearly make a look. Maybe there are some corona structures now to the molecule of the month. David Goodsell, he makes his fancy drawings and this kind of fancy contribution. There's a lot of educational stuff there present, so even when what I'm telling you is a bit boring. Yes, can be. Yes, look on this one. They have really very fancy resources there. It's really worthwhile to spend some time browsing over that. Not only to download structures, but also to look for the kind of resources they do have there. Visualize, analyze. The education stuff is really good. Yes, also newsletters whatsoever. So when you see how many structures, this is from last year, 150,000 structures. I have not looked today. I guess 178 or something like this. 1,000 structures. Most are from X-ray. The number of electron microscopy structures is strongly increasing now. Yes, because many large complexes are now made by electron microscopy. That number will increase. So it will certainly get up to, I suppose, 10-15% of all structures deposited will be in future now EM structures, and it may even around the number of NMR structures, because NMR structures take a lot of time to solve them. Yes, some hybrid structure and so on. Yes, that's something. Numbers are going up. You see here the growth. Yes, going up. At the beginning till about 2010, 11, it was exponential growth. It's not the corona growth curve here, which we do see. It's a growth curve from 1976, actually predicted by Richard Dickerson, yes, by this fancy formula. 1960 was the first protein structure, yes, that one from myoglobin. He evaluated, and it was sticking up to about 2010. It was really following, and now it's mostly linear, so there's not a strong further growth in the number of structures anymore, which are coming up in addition per year. That number has now become more or less now a linear growth. But at the beginning, we had this tremendous increase, and we will talk in the lecture why this has been the case, what actually the kind of progress has been made. But you should remember, sequence space here, non-redundant protein sequences, is by orders of magnitude higher than what is being possible to make with to solve by structure. So modeling sequences by available structures is becoming ever and ever more important task for doing many biological issues. And remember the coronavirus stuff, okay, that has been very quick because the labs were very quick to produce the first structures of COVID-19 components, But even without this one, the models would have been at the hand already from the beginning as the sequence became available. And with this, I come now to the very, very end. Just, to cool down a little bit. So structural biology, yes, is has really some history, yes, So it was Darcy Wentworth Thompson, yes, who recognized or rediscovered again, yes, actually the Greeks have already found them by themselves for many cases, that there are strict mathematical and geometrical relationships in nature existing. Here you see Nautilus, for example, and when you find this here, this is following a spiral, yes, Nautilus is following a spiral that is an Archimedean, I think it's an Archimedean spiral, yes, so with linearly decreasing curvature going on. It's describing the structure. And that can be exactly found in nature, and he gives in this book many examples that what we find in nature, even when it's on the macroscopic level, has to follow this kind of certain rules, and that can be explained from first principles, or here even in terms of evolution, yes, this kind of morphogenesis and morphogenetic alteration, yes, between different kind of species. Something similar we do also find on the microscopic level, with structures. And this is my last slide, yes, who comes to Jena, the only city without any kind of corona cases at the moment, can visit Ernst Haeckel's a museum. Maybe not, because it's now everything is closed, because Ernst Haeckel, he was already collecting structures of macrobiological structures, yes, here in this kind of sea animals, cataloging them and finding related motives. And next time we will talk about domains and motives. I thank you very much for your patience and attention, and hope that you could get something out of it. I have time for questions, but I will switch off now any kind of recording that is going here, I think, still.